In this video, we are going to talk about receptive fields in the visual system, particularly what they are and what purpose they serve in visual processing. Receptive fields are a fundamental functional unit of vision and the primary means through which visual information is detected and processed by the brain. They are located throughout the visual system and become more sophisticated and selective in higher cortical areas. Let's first talk about the most classic example of a receptive field, which can be found in the retina. Retinal ganglion cells are neurons that are responsible for transmitting light information to the brain for processing. There are approximately 1.2 million retinal ganglion cells, which synapse with approximately 120 million photoreceptors across the retina, with cones more centrally located and rods more prevalent in the periphery. To ensure maximum spatial resolution, you would normally have one photoreceptor synapsing with one ganglion cell. And this does occur within the fovea to ensure that this area can resolve maximum detail. However, there are substantially less retinal ganglion cells than photoreceptors, which poses a biological problem regarding how to relay information from many more photoreceptors than there are retinal ganglion cells. The solution to this problem is achieved by a retinal ganglion cell pulling information from a number of photoreceptors within a small spatial region through synapses with a collection of supporting bipolar and horizontal cells that form a localized cellular network. This spatial pooling forms a receptive field in which photoreceptors within the spatial region or field contributes information to the retinal ganglion cell. Receptive fields in the retina are circular in shape and are highly organized such that they exhibit a center-surround antagonistic arrangement. For example, an on-centered cell will respond when light stimulation occurs at the center of the receptive field by increasing the rate of neuronal firing or response. A light stimulation of the surround inhibits the cell by inhibiting neuronal response. Light over the entire receptive field results in only baseline activation as excitatory and inhibitory zones balance each other. Receptive fields have complementary arrangements and may be off-centered with light stimulation in the center resulting in response inhibition, but light stimulation in the surround resulting in neuronal excitation. Receptive fields thus work on the principle linear summation or pooling of light, and with the center and surround regions interacting in the process called lateral inhibition, in which adjacent regions of the receptive field may inhibit each other. Here, for this on-centered cell, initially as a spot of light increases in size, cellular activation increases consistent with spatial pooling, reaching maximum when the center is completely illuminated, but with increasing light illumination so that it encroaches on the surround, there is a gradual reduction in activation because of lateral inhibition. Light covering the entire receptive field results only in baseline response as excitatory and inhibitory regions are balanced. Receptive fields can also be color sensitive and tuned to color opponent red-green or blue-yellow pairs. Here, for a red-green receptive field, red light results in neural excitation, but green light results in inhibition. Red-green receptive fields can also have the opposite configuration, such that green light activates the cell, whilst red light inhibits the cell. Complementary blue-yellow receptive fields can also be found in the retina. Receptive fields can be found throughout the visual system, and whilst in pre-stride areas they exhibit a classic center and surround arrangement, receptive fields in striate and extrastriate areas are further specialized to be able to process different visual attributes such as orientation, motion, color, and stereopsis. For example, in the striate cortex, cells are known to be tuned to orientation and have receptive fields that are elongated and respond to a bar of light of a particular orientation placed in the center of the receptive field. Here, a bar of light that is vertical causes maximum excitation, but cellular response gradually reduces with orientation change. These orientation tune detectors are referred to as simple cells and are believed to be formed by pooling the response of a number of classic receptive fields along a particular spatial direction. In summary, receptive fields are the most basic functional unit of vision and represent a region of space over which a cell responds to light information. Receptive fields in lower cortical areas respond well to light stimulation, but become increasingly more complex and selective for global motion and object form in higher cortical areas.